Hello student, welcome to Anu's classes. The topic of my last video was double fertilization, but due to some editing problems the video was not clear, so today I am going to explain the same topic once again. So now proceed to the topic double fertilization. It is one of the most important topics. So double fertilization is the process where the fusion of two male gametes takes place with two different types of cell within the same female gametophyte. It means in this process at the same time one male gamete fuse with one different cell and another male gamete fuse with another cell within the same female gametophyte. Since two different types of fertilization takes place at the same time within the same female gametophyte, that's why the term double fertilization is given to this process. So now before going to the steps of double fertilization, you first go through this page. So this is the structure of a mature flower. These are the male organ and these are the female organ. The stalk of the male organ is known as filament and the uppermost head portion is known as anther, where the pollen grains are produced. These are the these are the female organ. So uppermost portion is called stigma, then style, and this is ovary, and ovules are arranged within the ovary like this. This is the annular structure of a ovule and this is the innermost structure of the female gametophyte. So when in the female gametophyte there are total 7 number of cells. These 3 cells are this side. These are as a whole known as egg apparatus. Means these 2 orange cells are known as synergies and this pink cell is known as egg cell. And as a whole these 2 type of cells together known as egg apparatus. And these 3 blue color cells are known as antipodal cell. In the center there is another cell which has 2 nuclei, that is known as secondary nucleus, and these nuclei are known as polar nuclei. So in this structure there are total 7 number of cells. This 3 plus 3 6 plus 1 7. But there are 8 nucleus. 1, 2, 3, 4, 5, 6 and 7, 8. That's why this structure is also known as 7-celled 8-nucleate state. So if question come, what do you mean by 7-celled 8-nucleate stage? Then you have to write about this. You have to write about this embryo sac. Now come to the process of double fertilization. So when the flower become mature, the mature pollen grains are deposited on the stigma by the process of the pollination. But at the same time, on the stigma, there are a lot of pollen grains are stick like this. But out of this pollen grain, only one, which is the most suitable one, is selected for the double fertilization process. As the pollen grain is selected, the stigma of the female organ release a liquid which has sucrose and water, and by absorbing that liquid, the pollen grain start germinating. And then it start forming a tube-like structure which is known as pollen tube. And by the help of this pollen tube, this pollen grain first penetrate the stigma, then style, and then it come to and reach the embryo sac like this. At the same time, the nucleus which are present within the pollen grain, one is called vegetative cell, another is called generative cell. They also move within the pollen tube. And in the meantime, these generative cells divided into 2 cells which are known as male gamete. It means the vegetative cell remains same, but the generative cell divided into 2 cells which are known as male gamete. Then the pollen tube grow with 2 male gamete and 1 vegetative cell and finally reach the embryo sac. And when it reach the embryo sac, the structure look like this. So when the pollen grain, tip of the pollen tube reach the wall of the embryo sac, the 2 synergies and 3 antipodal cells, these 5 cells start degenerating. And after that, the pollen tube penetrate the wall of the embryo sac like this, and it release 2 male gametes within the female gametophyte or the embryo sac. Then the first male gamete fuse with this cell and the second male gamete fuse with this cell. It means the first male gamete fused with the egg cell, and as a result it produced a twice-end structure which is known as zygote. Then the second male gamete fused with the secondary nucleus which already has 2 polar nuclei and finally form a thrice-end structure which is known as PEN, primary endosperm nucleus. So this first fertilization process means fusion of male gametes and egg cell is called syngamy. Syn means 2, gamy means gamete. It means in this process fusion of two nuclei takes place, that's why it is called, or two gametes takes place, that's why it is called syngamy. But here fusion of three nuclei takes place, that's why it is called triple fusion. And till then the antipodal cells will be completely degenerated. Then after that, they form a structure like this, two nucleated state structure, one three nucleated state structure. That this is called zygote and this is called pen, and this process is called syngamy, and this process is called triple fusion. It means triple fusion plus syngamy is equal to double fertilization. So this is the whole process of double fertilization. Now go to the flow chart of double fertilization so that you can easily remember the topic. First male gamete fuse with the egg cell. As a result, they produce zygote, which later develop into the embryo, and this process is called syngamy. Second flow chart, second point, second male gamete plus polar nuclei. Polar nuclei means in place of polar nuclei you can write here also secondary nucleus, and if you write polar nuclei, then nucleus means two polar nuclei, and it from the primary endosperm nucleus means pen. And it is a trisome structure, and process is called triple fusion, and this syngamy plus triple fusion is equal to double fertilization. Then next point, the zygote developed into embryo, the pen developed into endosperm. And what is the function of endosperm? Provide nourishment to the developing embryo. Next point, the pollen at the when during pollination stage, it was two cell stage, one is generative cell, one is vegetative cell. Then on pollen tube it become three cell stage, one vegetative cell, and the generative cells are divided into two types. One is Z1 cell, another is Z2 cell. Or the Z1 cell fuse with the egg, and as a result zygote is formed, and the Z2 cell fuse with the polar nuclei, and as a result it produces pen. So this is the total process of double fertilization. Thank you.